Ebola, Marburg. Mysterious viruses that rip bodies apart from the inside out and kill up to 9 of every 10 people they infect. What will happen when these microscopic assassins come to your town? The story you're about to see is fictional, but the science is real. Imagine that it's the day after tomorrow, somewhere over the Ugandan jungle.
2 rebels were in this area. The captain came around the tree.
The United States government has sent a doctor here to solve a medical murder mystery. If he succeeds, he could stop a killer before more victims suffer and die. Like all detectives, he follows a trail, looking for clues in the recent past.
Delta 2 and 3, this is Delta 1.
I'd like to know what's going on. Am I infected?
48 hours earlier, at a military base outside of Washington, D.C., an American Special Forces officer returns from a mission in Uganda. He travels light, just his rucksack and his guns. He's an intimidating man. But the most frightening thing about him lies underneath the skin. A virus has entered his body. Right now, it's down in the thin capillary walls, nearly lost among the discs of hemoglobin. A strand of Ebola. Only 80 nanometers wide, the Ebola virus is inconceivably small, but it causes enormous damage. Its worm-like form contains a piece of genetic code that forces the human body to self-destruct. After months of fighting in the jungle and a long flight home, the soldier thinks his trials have come to an end. In fact, over the course of the next 48 hours, things will get much, much worse.
Welcome back, sir.
Hey,
How's it going, soldier?
when'd you get back?
Just today.
Yeah? What happened to your hand?
You should see the other guy.
Let's go get some beers tonight. What do you think?
Sounds great.
All right.
The exhausted soldier mistakes his fatigue for jet lag from the long flight back to the States. This is the first stage of a biological meltdown. Ebola forms half of a very small disease family called the filoviruses. Marburg virus is the other member of the clan. Both cause viral hemorrhagic fevers, probably the most frightening of all diseases. Marburg and Ebola tear you apart from the inside, reducing your body to a sack of blood and jellied tissue. They're also unusually lethal. Killer diseases such as the plague and yellow fever take the lives of about 20% of the people they infect. The fearsome smallpox virus has a 30% mortality rate. Filoviruses kill on the order of 50 to 90%. So far, Ebola and Marburg have mostly predated upon the people of Central Africa. It seems that both viruses lurk somewhere in the jungle, and that incursions into the rainforest have brought them out of hiding. Scientists suspect that the filoviruses emerged thousands of years ago. Virologist Peter Jarling has studied Ebola and Marburg for more than a decade.
There are no other viruses in nature like it. It seems like the primordial virus that must have been around a long time because there's nothing else that it might have evolved from. There's been speculation that some of the biblical plagues and what have you might in fact have been Ebola.
They may be ancient, but until recently the human race had no idea filoviruses exist. Marburg was discovered in 1967, when a batch of research monkeys infected laboratory workers in Yugoslavia and Marburg, Germany. As is the custom with viruses, the disease was named after the place where the first outbreak occurred. Ebola slithered into view in 1976, killing 280 of the 318 people it infected. Ebola Zaire, named after a river in the country now known as the Democratic Republic of Congo, remains the deadliest known strain of the virus. After disappearing for 20 years, it resurfaced in the mid-90s and continues to decimate African towns every few years. 3 other strains of Ebola have also been identified: Ebola Sudan, Ebola Ivory Coast, and Ebola Reston. Together, they have infected and killed nearly 2,000 people. Marburg was once considered the lesser threat of the filoviruses. Recently, it has proven just as deadly as its cousin. At the turn of the 21st century, a Marburg outbreak among miners in the Congo killed 128 of the 154 young men it infected. Another Marburg epidemic that began late in 2004 killed over 300 people in Angola, many of them children. No one knows how these epidemics started. In fact, for many months, the people of Angola didn't even realize that Marburg was on the loose. Most of the early cases were misdiagnosed as malaria, typhoid, or yellow fever. So far, Marburg and Ebola epidemics have been confined to Central Africa. Outbreaks outside of Africa were the result of laboratory accidents. Sooner or later, though, someone will carry these diseases to the city, and then to the rest of the world. hours. The officer has a visitor.
I figured you might not be busy. Welcome home.
Welcome, yes, it is.
Like HIV, Ebola spreads through an exchange of bodily fluids. Even a cough can prove fatal for someone hit by a few droplets of spittle. So intimate contact with an Ebola victim goes well past the boundaries of safe sex. As the soldier sleeps, the virus gains footholds throughout his body. Ebola Zaire's week-long incubation period is over. (0800 hours). The returning soldier has been back in the United States for less than a day. The Ebola virus he carried back from Africa replicates at frightening speed. The first signs of Ebola are a severe headache, nausea, and pain throughout the body. The soldier knows he's sick, but at this point he can't think clearly. High fever and exhaustion have taken over. He may think he has malaria or typhoid, or the worst hangover of his life. His blood won't coagulate properly. Blood leaks from his nose, mouth, and eyes. Eventually, it will stream out of every opening in his body. These agonies are just side effects of the real problem. His internal organs are ripping apart. Inside his liver, small fissures open up. The slender capillaries that carry blood through the body are tearing. As the capillary walls open, blood seeps out. This happens across his liver. Soon the hemorrhaging will spread through his body. As the infection eats into his brain, his lethargy and disorientation will only get worse. When mysterious diseases fell soldiers, the US Army Medical Institute of Infectious Diseases finds the cause, and sometimes the cure. USAMRIID sits 50 miles outside of Washington, D.C. Until 1969, this was the heart of America's biological weapons program. Now, the well-guarded facility performs the opposite mission. It's where doctors battle the most dangerous pathogens on Earth. American soldiers see action around the world. USAMRIID's mission is to protect them, and everyone else, from the diseases they encounter along the way. The dangers of studying microkillers like Ebola can't be overstated. If all the diseases studied here escape, their combined destructive force might be impossible to stop. The laboratories at USAMRIID are designed to make sure that nightmare never happens. Labs break down into 4 zones of biological safety. The lowest biosafety levels, BSL 1 and 2, handle treatable or non-lethal agents such as measles, mumps, and the rhinoviruses that cause the common cold. Biosafety Level 3 labs study organisms that cause serious or fatal diseases, but for which vaccines or treatment is available, such as plague, rabies, and tuberculosis. A one-way negative pressure airflow keeps dangerous germs trapped inside. In BSL 3, specialists must always be gowned and gloved. And then there is Biosafety Level 4, a level so hazardous that the air and water running through the lab is continuously pumped out and superheated to kill all the germs. BSL 4 handles hot viruses, living specimens of pathogens like smallpox, anthrax, and Ebola. Highly infectious killer diseases for which there are no treatments and no cures. Researchers brave enough to work here get dozens of inoculations and months of training before they're allowed through the airlocked doors. They labor in full-body spacesuits with independent air supplies. Working in this lab requires nerves of steel. A slip of a knife or a needle could mean exposure and agonizing death. USAMRIID's senior research scientist, Dr. Peter Jarling, knows the dangers of studying Ebola firsthand.
There are no treatments available, there are no vaccines, there is no countermeasure that was shown to be effective in treating Ebola, so working with it is to recognize that if you catch your experiment, you've got a pretty good chance of dying.
To keep that from happening, BSL-4 workers follow a strict procedure of decontamination and protection. First, they strip down and go through a series of airlocks. There's a shower before they grab their clean scrubs and put on their spacesuits. Purple scrubs are worn going in. They'll be autoclaved on the way out. The airtight spacesuits are made of thick polyethylene plastic. Once he's inside the suit, purified air pumped through a hose keeps this technician alive. If he's disconnected, he has 4 minutes maximum to connect to another air hose. After that, it's a choice between suffocation or unzipping the suit and letting in whatever floats in the air. Next, there's one more shower, a hot spray of ammonia. When his shift is over, this soldier must repeat this entire procedure in reverse. Peter Jarling has spent the better part of his career working at Level 4.
Working in a spacesuit is certainly a tough way to make a living. The greatest inconvenience is the rather loud rush of air, which is persistent. It makes it very difficult to communicate, makes it impossible to talk on the telephone. It's also very dry air, so you tend to dehydrate, but you don't want to drink too much fluid for obvious reasons. One has to be aware of where the air lines are so they don't get entangled with other workers in the laboratory or get wrapped around laboratory equipment. So it's very cumbersome and frankly quite strenuous.
Medical research means working with knives and needles. Dangerous to begin with, but vastly more so in a lab filled with Level 4 microkillers. Since it opened in 1969, no one at USAMRIID has died from exposure to any infectious agent. But there have been about 20 accidental exposures to lethal diseases, including Ebola. An accident in BSL-4 results in a long-term stay in the Slammer, a Level 4 holding cell locked from the outside. For decades, few people outside of the military knew about USAMRIID's high-risk medical mission. Then Ebola came to America.
I actually never worked on Ebola virus myself until it fell into our lap during the Ebola Reston outbreak. In fact, Ebola virus was the one virus I didn't want to work with because it scared me more than everything else combined.
In 1989, a breeding farm in the Philippines sent 100 monkeys to the United States. They ended up in a medical research laboratory in Reston, Virginia, just outside Washington, D.C. Soon, the monkeys began to die for no apparent reason. The puzzled lab sent blood and tissue from the dead monkeys to USAMRIID. Peter Jarling tested the mysterious samples. He found himself looking at a new strain of Ebola. Jarling ran his findings over to C.J. Peters, then USAMRIID's chief of disease assessment. Peters was gripped by a feeling he calls the pucker factor.
Major pucker factor was when Peter Jarling pulled out that electron micrograph with Ebola virions. And he said, well, we just cultured this out of a monkey 30 miles from here in Reston, Virginia. And I'll admit, the hair stood up on the back of my neck. You don't like to think about these things as being near home.
A Level 4 pathogen lurked in the suburbs of America's capital city.
Here's a virus that looks like one of the most lethal viruses we know about. It's killing monkeys and spreading among the monkeys. What's going to happen if a human gets infected with that virus? Well, we didn't actually want to find out, so we recommended that they depopulate the monkey house.
The laboratory killed the infected monkeys. Then monkeys kept down the hall from the first batch started dying. Frightening news. It meant that the new strain of Ebola traveled through the air. Ebola Reston killed 100% of the animals it infected. If it escaped the building, and if it infected humans, the possibility was too horrifying to consider. Peters sent in a USAMRIID team to purge the monkeys and clean out the lab. These rare pictures show the small army medical unit that risked their lives to clean up the hot zone. After killing all the monkeys in the building, the team scrubbed every surface of the lab with bleach. Then they burned formaldehyde for 3 days to kill any remaining trace of the virus. It wasn't over. 4 laboratory workers tested positive for Ebola infection.
There were 4 human infections. None of those human infections was symptomatic. So in one of those dodged a bullet minutes, we found out that that virus was much less pathogenic for humans than the classical Ebola strains.
The first outbreak of Ebola Reston was contained, but the virus itself is still on the loose. In 1992, Ebola Reston struck a research facility. Then in 1996, the virus killed more research monkeys in Texas. Both outbreaks were tracked back to the Philippines. How did a disease that lives in Africa infect monkeys in Southeast Asia? Investigators never learned the truth. Shippers and suppliers in the monkey trade proved reluctant to share information. But in a desperate bid to stop Ebola's deadly spread, all the monkeys of the Philippine breeding farm were euthanized.
Yes, I consider them as children also. These babies. I think you are going to kill it. Oh, God. Crazy.
Killing infected and at-risk populations stops outbreaks. But what happens when the disease moves to humans? Day 3, 1500 hours. The returning soldier has slept for more than a day.
Diamond! Open up! Diamond! Diamond, I know you're in there. Open up the door. Come on, you blew me off last night. Wake up! Diamond, look at me. Look at me. Do you know where you are? Diamond, look at me. Look at me. What happened? Was this while you were gone?
He has no idea what's wrong, and though logic demands he keep his distance, his gut response is to help a sick brother in arms. But no good deed goes unpunished. As the infected soldier bleeds, he sheds millions of Ebola viruses.
When you touch an Ebola patient, you're only a few microns away from a pool of infectious virus.
Come on.
And if there are any tiny skin breaks, or if the patient has bled onto an area of skin, you're intensively exposed to infectious virus.
Come on, Diamond. We've got to get you to the hospital, man. Come on, it's time to go. Let's go.
Stay with me. Come on. Stay with me. Stay with me. Come on. Just hold on to me. Hold on to the rail. Come on, buddy. Good. Hold on. Come on.
Yeah, you looking for Megan Close?
1500 hours. Time has run out.
Nurse! Hey, I need a nurse! Hey, nurse!
The Ebola virus burned through this soldier's body in a matter of days.
Help me out! Help him! Do something to help him! Don't just let him lay
Inside, massive hemorrhaging has broken down the tissue of the soldier's major organs. Autopsies on Ebola victims are often frustrating affairs. The internal damage is so extensive that there's little left to study.
We can't do anything for him right now. We can't help till the recon team shows up. Just head off the floor.
There are many paths into the bloodstream. For instance, the thin capillaries of the eyes. Once the worm-like strands of Ebola get inside a new host, no one can stop them. Infection and death may soon follow.
All right, I got him. I got his head.
Just make sure that it's around more.
Okay, yeah, I got it.
1 milliliter of infected human blood can contain between 10,000 and 1 million pieces of Ebola virus. Doctors, particularly on a military base, would immediately suspect a hemorrhagic virus. Their first step, isolate anyone exposed to the virus, decontaminate them with showers, then confine them for weeks of testing and observation. Few welcome this prospect. But decontamination and isolation are essential parts of the strategy to contain an Ebola outbreak. Hospitals in wealthy nations have the equipment and training to handle this kind of health disaster. But the situation in rural African hospitals is dire. Maintaining sterile conditions here is extremely difficult. Money is scarce, and sometimes clinics cut costs by reusing needles, a sure way to pass Ebola infections from one patient to another. Ebola victims are often crowded into wards or surrounded by their unmasked, unprotected loved ones. This makes it easy for the virus to spread to visitors. Hospital workers also fall prey to the virus and help spread it to other patients. Epidemiologists call this amplification, when poor medical care turns an outbreak into an epidemic.
These Ebola epidemics generally start out with one case. They get sick in a town or a village. They spread the virus to the people who take care of them. Eventually somebody goes into the hospital. And in an African hospital where there are no gloves, no gowns, no masks, no precautions are taken, you have a perfect setting for the transmission of this virus. The disease will continue to be transmitted until literally everyone in the hospital dies or leaves.
It shouldn't come as much of a surprise then that healthcare workers are often shunned and attacked during epidemics. Villagers see that many of the sick people who go into hospitals don't come out alive. Some believe that the clinics deliberately spread Ebola and Marburg to increase their business. Cultural factors don't help either. In Central Africa, the white garb worn by doctors is considered a symbol of witchcraft. These fears have led people to hide their sick and dying relatives from rescue workers. The problems don't stop when the victims die. The virus can survive for weeks in a dead body, and corpses teem with Ebola. The ceremonial handling of the dead is a major cause of infection. Ebola is a vicious disease anywhere, at any time. But the conditions in impoverished regions make it that much worse.
It's got a lot going for it in terms of trying to adapt to humans. And I think it's very dangerous to let these epidemics run in Africa, as often happens because resources are not brought to bear, social disruption prevents our intervention.
If doctors understand what they're dealing with, they'll immediately act to contain the outbreak.
What's the matter with me? Am I infected or not? Hey, I know you're out there.
If our scenario comes true, the soldier who'd been exposed to Ebola will end up locked in a level 4 slammer. He will be kept here, isolated and under constant observation, until he is declared healthy or until he dies. Meanwhile, BSL-4 doctors will analyze his blood. This sample will be put under an electron microscope, a camera powerful enough to photograph atoms and viruses. And this is what they find, a particle of Ebola Zaire. While the lab looks deep inside the virus, epidemic investigators trace the infected soldier's path. Like crime scene investigators, they look for clues to solve a biological murder mystery. Where did he get the disease? Was this contamination accidental or deliberate? In other words, was the soldier a victim of biological warfare? Or was he somehow exposed to Ebola in nature? How long was he sick? Did he spread the virus to others? With Ebola, every second counts. The source of the infection must be quickly found and contained. That's easier said than done. The longer it takes to learn the origins of an outbreak, the greater the chance it will grow into a massive epidemic. Fortunately, the soldiers' movements were restricted to a military base. In this tightly controlled environment, it's relatively easy to lock down and decontaminate the entire area. Meanwhile, everyone the returning soldier had contact with must be tracked down and put under observation until they test negative for the virus.
Said he would survive. Ever. Does anybody survive?
The odds are not good for the soldier's girlfriend. The Ebola virus has taken root in her liver. Despite decades of research, much about Ebola remains a mystery. It's thought that the disease sabotages the body's defenses by invading and killing the white blood cells that fight off infections. The virus evades immune system attacks by stowing away inside the bodies of macrophages, the patrol officers of the immune system. Macrophages clear out bacterial infections, but they can be vulnerable to viruses. A macrophage does not die quietly. It sends out a red alert, madly spraying the chemical cytokine as it's carried along the bloodstream. At normal levels, a cytokine burst warns the rest of the immune alert system to come and join the fight. But when Ebola attacks, something goes haywire. The cytokine bursts tear open the blood vessel walls. Blood leaks into the surrounding tissue. This cycle repeats until the victim bleeds out. No one yet understands exactly why this happens. But we do know that when the process begins, the massive hemorrhaging is nearly impossible to reverse.
Really just a measure of diffuse organ damage. It's kind of like the death of 1,000 cuts. You have these many, many tiny lesions throughout the body.
In the US, the goal is to stop the virus before it claims more victims. Meanwhile, a medical investigator travels to Uganda, searching for the source of the outbreak. The virus hunter comes armed with a detailed knowledge of how diseases spread and a cluster of facts about the case. He knows that the infected captain went out on one last patrol before going stateside. The captain got the virus, but the two sergeants traveling with him did not. Why? He must also determine whether this is an isolated incident or part of a larger epidemic that so far has gone unnoticed.
This was the first village where we had the occurrence, right there in the Kolo.
The trail is now a week old, the doctor has to act while there are still clues to follow.
Occurrences all over. But here is the main problem area. Up to Moon
Excuse
to
me,
way
sir.
down south.
That's actually the area where we found the rebels, Cat Zero, dead.
Was there much blood? Any bleeding?
Yes, sir, 2 to the chest. Definite rebel attack.
I need to know exactly where he went. Retrace every step you guys took that whole day.
Yes, sir.
When virus hunters have been able to identify the first victim of an Ebola outbreak, that person has often had some sort of exposure to the dense core of the African rainforest. Once, this vast jungle was nearly impenetrable, but as populations expand and resources dwindle, more and more humans are making their way into the primary and secondary forests. The doctor has come here with 2 goals. First, to reconstruct the sequence of events that led to the captain's Ebola infection. Second, to chase every virus hunter's dream, finding Ebola's elusive hiding place in nature, what virologists call its reservoir. A reservoir is an animal, insect, or plant that carries a virus but doesn't get sick. The problems start when life forms without immunity get infected. In the case of Ebola, the natural reservoir, which is probably an animal, somehow passes the virus to primates. Monkeys in particular succumb quickly to infection. They can pass the Ebola virus onto humans who have similar biochemistry. Locating Ebola's reservoir will help doctors study the disease's life cycle and perhaps find ways to stop it. But so far, their efforts have been stymied by the sheer size and biodiversity of the jungle.
Ebola virus is certainly one of the most fascinating viruses. There's an elusive quality about the virus. Its reservoir in nature has remained a mystery for all these years, despite some very intensive efforts to go into areas where disease has occurred and to trap everything that crawls, walks, and flies. All of those heroic efforts have met with failure.
In 1995, Ebola Zaire resurfaced and killed 250 people. As head of the Centers for Disease Control's Special Pathogens Unit, C.J. Peters led an extensive search for Ebola's hiding place.
We were able to trace back to what we believe was the original infection, the man who initiated it, the famous patient 0. So we thought, well, that's easy. We know who it was, we know what this guy does, and we will just do studies around his history. Turns out, this guy lived in town. He rode a bicycle through grasslands to an area of secondary growth forest. And then, to make it even worse, a couple of days a week, he would ride his bicycle deep into primary forest, and he would sometimes even spend the night there. So he was exposed during the day and at night in the primary forest. So in essence, we had to look at everything. And when you start looking at everything in the tropics, you're in trouble. We processed all the mammals that we trapped and about 30,000 arthropods that we took, and we didn't find any trace of Ebola virus at all. The reservoir still remains a mystery.
Nobody knows for sure, but one animal fits the profile of an Ebola reservoir. Bats are migratory species, common enough to interact with humans, but not so common that Ebola would frequently break out. They could sustain the virus. Bats also arouse suspicion because several Ebola outbreaks have been tied to caves. It's possible that contaminated bat guano helps to spread the disease. Like all detectives, virus hunters follow trails and put together puzzle pieces. This investigator knows that the infected soldier was not near a cave, but he did come home with a cut across his hand. How did it get there?
Right where he is where the rebels were. That's where the captain got cut.
Delta 2 and 3, this is Delta 1. One hostile moving towards you.
A small flesh wound, nothing a combat veteran would think about twice. But the cut is big enough for a microscopic particle of Ebola to slip into the bloodstream. Now the virus hunter must find out how and why that happened, and whether anyone else was exposed. Three American soldiers in the Ugandan jungle, ready for anything that comes their way. Or so they think. The most dangerous enemy here is something they can't see. Something they can't fight. A thing that lurks in blood. The Ebola virus survives in the skin and sweat glands of victims long after their death. Any virus in this blood is probably inactive, but it can still be detected. The doctor suspects this is how the soldier was infected. When he analyzes the sample, he'll have proof. The soldier contracted Ebola through open wound contact with the blood of an infected corpse.
Bad idea.
That means Ebola Zaire is on the loose, and many local deaths misdiagnosed as typhoid and malaria may in fact be early cases of an Ebola epidemic.
And the villagers don't really understand what is going on. They think it's malaria, and it is not helping. It's only prolonging the illness.
Your first concern is to control the outbreak. But often you can't do that because many of these viruses are so poorly understood. You want to find out more about the disease to enhance your ability to control it, to enhance your ability to prevent the emergence of the disease in the future.
The biggest problem area though, and I think the man was
But another question remains, a question the army would ask that a civilian might not. Was this bad luck? Or was it an act of war? The doctor must consider the facts. US Special Forces support the Ugandan government's fight against a rebel army. The rebels hide deep in the forest, the same place Ebola lives. Suppose the rebels found that one of their men was infected. Would they take him to a hospital? Or would they shoot him and leave the infected corpse for the enemy to find, as a kind of poor man's biological warfare? They wouldn't be the first to think of using Ebola as a weapon. At the University of Texas in Galveston, C.J. Peters now heads a state-of-the-art center for biodefense and emerging infections.
But many of the bioterrorist agents are indeed emerging infections, are serious problems in nature. You don't just make viruses, you got to get a virus somewhere.
State-supported biological weapons were outlawed by treaty in 1972. But the Soviet Union kept at it. By 1991, they created a strain of Marburg virus that spreads through the air. They manufactured Marburg with the intent of loading the virus into missile warheads aimed at major cities across the United States. What happened to that supply? Supposedly, most of it was destroyed. But in the chaos that followed the collapse of the Soviet Union, it's impossible to know. Fortunately, there is some good news. Since the Reston outbreak of 1989, Peter Jarling and his colleagues have worked steadily on an Ebola vaccine.
There are various strategies for producing viral vaccines. The simplest is to simply grow the virus in quantity and inactivate it in some way, and use that inactivated virus as a vaccine. We tried that approach with Ebola virus, and although it protected small rodent models, mice, and guinea pigs, it failed when we tried it in monkeys. So I'd say we have a lot of dead ends, about 10 different strategies that should have worked but didn't.
But after years of failure, researchers have created a vaccine that immunizes monkeys and which may protect humans. The vaccine piggybacks inactive Ebola onto the virus that causes the common cold. The science is complex, but this technique could pave the way to vaccines for other seemingly unstoppable pathogens, such as HIV. If all goes well, soldiers and civilians may eventually have some protection from Ebola. But until then,
you like to think about these things as happening somewhere a long way away, where it's going to be very hard for it to get to an airport and get to where you are. Unfortunately, the way things are in the world today, all those places way far away are actually a lot closer than most people realize.
we can only hope that if we are infected, we will be one of the lucky few whose immune systems can fight off this terrible killer.